Good morning, students. I am Dr. Maitri Shah, Head Department of Botany, VPM's B.N. Bandorkar College of Science, Thane. My SYBSC students, I'll be teaching chromosomal aberration. This is a topic which is included in the course code USBO 302 in Unit 2. First, I'd like to define chromosomal aberration. Students, all of you know that chromosomes are the structure with a definite organization. Any variation in the structure and the number of chromosome is called as chromosomal aberration. So if you have to define chromosomal aberration, we define it as variation in the normal structure or the number of chromosome is known as chromosomal aberration. So chromosomal aberration is any irregularities or abnormalities in the structure of the chromosome, the arrangement of the chromosome, the number of the chromosome, and all these leads to chromosomal aberration. In your syllabus, they have included 4 types of chromosomal aberration, namely deletion, duplication, inversion, and translocation. All the 4 common types of chromosomal aberration which has been included in your syllabus are due to chromosomal aberration related to structure. So these are the irregularities in the structure of the chromosome that leads to these kinds of chromosomal aberration. So I will repeat it. What are the 4 types of chromosomal aberration? Deletion, which is also called as deficiency, duplication, inversion, and translocation. Definition of deletion. So deletion is a type of chromosomal aberration. So what happens in deletion? A small portion of the chromosome or a small segment of the chromosome or a small part of the chromosome gets deleted. When this happens, then in that case, it causes chromosomal aberration. So, students, deletion involves the loss of a part, a segment of the chromosome. So now we have to tell from which part. So what is the position of the deletion? So there are two types of deletion based on the position. So one is called as the interstitial deletion and the second one is called as the terminal deletion. Why does the deletion occur? There are many causes. So sometimes it originates spontaneously. We don't know the reason why it has occurred. Or sometimes it is artificially induced for various experiments. So deletion may originate spontaneously or it can be induced artificially. So let us study the different types of deletion. The first type is terminal deletion, made up of 2 words, terminal and deletion. You know the definition of deletion. So, and what is terminal? Terminal is a position. So, terminal position. The second type is interstitial deletion. Interstitial means in middle. So, it is somewhere in the middle or somewhere in between. It is also called as intercalary deletion. Again, made up of 2 words, interstitial and deletion. You know what is deletion, and interstitial is the position. You can also see I have mentioned a third type of deletion, students. This is called as microdeletion. Microdeletion is a teeny, very small portion of amount of the segment of the chromosome which is deleted. If it is deleted, then it is called as microdeletion. What happens over here? In this kind of deletion, only few genes are deleted. So when there is a loss of a tiny segment of the chromosome, then in that case, it is called as microdeletion. Now this tiny deletion, like it was too small to be seen through light microscope or by normal karyotyping. However, now we have found out that it occurs randomly. And some of the examples which you can study under microdeletion is Dijudge syndrome, Angelman syndrome. So these are the syndromes which happens if there is a microdeletion. We will study in detail terminal deletion. As I told you, terminal deletion takes place because the segment which is deleted is towards the terminal end. Let us look at this diagram in front of us. In this diagram, you can see a normal chromosome. This normal chromosome has got the P arm, which is the short arm, the centromere, and the long arm, which is also called as the Q arm. So this is a normal chromosome. Now let us see what are the genes present on this chromosome. In the P arm or the short arm, you can see the gene is A and B. Then you can see the centromere, and then in the Q arm or the long arm, you can see the gene C, D, E, F, G, and H. So this is a normal chromosome. Now what happens in this chromosome? You can see deletion. Deletion takes place. Now let us study what kind of deletion takes place. So if you observe the P arm or the short arm, so you can see there is no loss of gene. Both the gene, that is A and B, is present. Good. Let us come down and you can see over here the centromere is present. Now let us look at the Q arm or the long arm. In the long arm, the gene C is present, D, E, F, G. What has happened to the H gene? So you can see a small segment is missing, or a small segment has disappeared. So in this chromosome, deletion has taken place and a small segment is lost. So that's why it is an example of deletion. From where the segment is lost, what is the position? The position is terminal. And because a terminal position, from the terminal position, a small segment is lost. So that's why it is called as terminal deletion. So you can see the lost portion over here, a small segment, and it is written lost. So why is it written lost over here? It has got the H gene because it has been seen that if centromere is not present in the next generation, that is after replication, you will find that this particular segment is lost. Centromere has to be present for a chromosome to survive. Now, just to repeat, terminal deletion, we can say that deletion is terminal when a single break occurs at the end of the chromosome. Now, what is important over here is that the word single break. A single break occurs. So when a single break occurs at the end of the chromosome, then what happens? Due to one break, the end of the chromosome is lost. So due to one break, a single break, the terminal end of the chromosome is lost. And this is called as terminal deletion. Terminal deletion is usually lethal. It depends if the size is big, the segment size is big, then there are large number of genes which are lost, and that can cause death of an organism. If the deletion is small and it involves only few genes, then in that case we can say that the organism can survive. Sometimes they survive in spite of having deficiency. I'd like to represent the terminal deletion with the help of line diagram. So what is a line diagram? So students, if you can see this carefully, you can see there is a line which is drawn, and this line represents a chromosome. On the line, you will see I have written few numbers. It is from 1 to 9. So it is 1, 2, 3, 4, and then you can see a circle which is colored, blue color circle, which is the centromere. And then 5, 6, 7, 8, and 9, which are the genes. So this is the line diagram of a normal chromosome. Now let us go to the second line diagram. So what happens in the second line diagram? What have I drawn? I have drawn a single break. Now you can see the single break is towards the terminal end of the chromosome. So I can say this is the Q arm of the chromosome, and towards the terminal end you can see the break. Now let us see what happens. Because of the break, the small segment which is carrying the gene 8 and 9 is lost. So you can see in the third diagram, I have the chromosome, and after the 7th gene, you can see the small segment is drawn below, and it is represented with the gene 8 and 9, and besides that, what is written? Lost. Lost because centromere is not present. Now, the next line diagram. So this is the resulting chromosome which is showing terminal deletion. What do we have here? We have the chromosome with the gene 1, 2, 3, 4, centromere 5, 6, and 7. What is lost is 8 and 9, the terminal segment. This line diagram shows terminal deletion. I hope students you have understood this line diagram. Okay, so now let me go forward. Let me study the second type of deletion. The second type of deletion, we call it as intercalary deletion, or we also call it as the interstitial deletion. Intercalary deletion requires 2 breaks in the chromosome. So in terminal deletion, I told you 1 break. Now for intercalary deletion, I'm telling you that for intercalary deletion, we require 2 breaks. Now let us observe this diagram. What is there in this diagram? In this diagram, you can see one normal chromosome. And in this normal chromosome, what are the genes you can see? You can see the genes A, B, C, D, E, and then you can see the centromere. Okay, and after the centromere, you can see F, G, and H. So what is A to H? A to H are the genes. Now, you can see a small portion is colored, and this portion, in this portion, you can see the gene D is present. What did I say? For intercalary deletion, you require 2 breaks in the chromosome. 1 break occurs over here, and the second break occurs over here. And when that happens, the small portion gets deleted. So what is the resulting chromosome? The resulting chromosome has the gene A, B, C, E, centromere, F, G, H. What is lost? A small segment carrying the gene D. So due to 2 breaks at interstitial segment of chromosome is lost in intercalary deletion. So let me come to the line diagram. It's a very easy way to understand deletion. So this is my normal chromosome. Now this normal chromosome, as you can see, I have drawn a line, and this line represents the chromosome. Students, here you can see there are, I have written 1, 2, 3, 4, 5, 6, 7, 8, and 9. So what are these? These are the genes. So what are these? Again, these are the genes. And you can see one blue color circle, that is the centromere. Now, the second line diagram, again you can see I have drawn breaks. But here there are two breaks, the first break and the second break. So if you observe carefully, you will see that the line is the chromosome 1, 2, 3, 4 gene. Then we have the centromere. Then between 5 and 6, there is one break. Then we have the second break between 7 and 8. So there are two breaks in the Q arm of the chromosome. What happens when there is two break in the Q arm of the chromosome, as in this example? What happens over here? The small intercalary segment carrying the gene 6 and 7, it is lost or it detaches from the chromosome. And after it detaches, the 5 end and the 8 end, it attaches, and you can see the chromosome formation, the one which is formed, it has got the gene 1, 2, 3, 4, centromere, then 5, and then 8 and 9. So what is lost is 6 and 7. A small segment carrying the gene 6 and 7 intercalary in position is lost because there were two breaks. One break between 5 and 6 and the second break between 7 and 8. So this is an example of interstitial deletion. So students, from this line diagram, I hope you could understand that how does the terminal deletion takes place and how does the interstitial deletion takes place. In terminal deletion, the position is terminal. It requires only one break. In interstitial deletion, the position is intercalary. It requires two break. In both the cases, a small segment is lost, and in the segment there are few genes are present, so few genes are also lost due to deletion. Now what happens at the time of synapsis? What is synapsis? Students, all of you know synapsis or pairing of chromosome. It takes place, and like in prophase 1, all of you have studied, synapsis takes place in the subphases where we have studied leptotene, zygotene, so pachytene. So in pachytene, you know that exchange of chromosome takes place. So synapsis is an important phenomena. What happens if there is an interstitial chromosomal aberration or an interstitial deletion? Then how does the chromosome pairing takes place? Now, what happens is, suppose it is a homozygous deletion, then in that case, the pairing or the synapsis appears normal. I will be explaining the same thing with a line diagram, but let me say what happens if the deletion is heterozygous. If it is heterozygous, then in that case, there is a presence of deletion loop. So now students, I have to explain to you what is a deletion loop. I feel that you know what is synapsis, you know what is homozygous, you know what is deletion, you know what is heterozygous, you know what is homologous chromosome. Okay, now let me go forward and let me come here, and I think I take it for granted that you know what is a homologous chromosome, but still I'd like to show it to you. Just look here, the line diagram which is in front of you. I have drawn two lines and they are of different color, and you can see that they are carrying the same gene 1, 2, 3, 4. Then they have the centromere, and followed by 5, 6, 7, 8, and 9. This is our homologous chromosome. Now, in this homologous chromosome, what happens is that we find that what happens, a heterozygous deletion has taken place. Now let us study what is heterozygous deletion. I have told about you the earlier slide. I mentioned heterozygous deletion. So in a homologous chromosome, what happens is that only one chromosome shows deletion, interstitial deletion, where the other chromosome of the homologous chromosome, it remains normal. So in the second chromosome, the deletion has not taken place. Then in that case, it is called as heterozygous deletion. So let us see in the heterozygous deletion with the help of line diagram. So you can see in this line, the chromosome has got the gene 1, 2, 3, 4, centromere 5, and then 8 and 9. So student, what is lost? Where a segment is lost? Is it? Yes, a segment is lost. And it carries the gene 6 and 7. So you can see that it is shorter in size. Why is it short? Because it has lost an intercalary segment carrying the gene 6 and 7. The second chromosome, the pair, you can see it is normal. It has got all the gene and the centromere is intact. So now synapsis has taken place. So at the time of chromosome pairing, so it will not pair because till 5 it is pairing or it is matching. But afterwards you have the gene 8 over here, but the gene 6 over here, 7 against 9, whereas 8 and 9 is over here. So it has to match at the time of synapsis. So what happens? To match each other, the portion between 5 and 8 in the normal chromosome, it forms a loop-like structure. And in this loop-like structure, the gene 6 and 7, it is present. So what happens is, because the chromosome showing deletion is shorter in size, to adjust with this and to match the genes to each other, the normal chromosome forms a deletion loop. So whenever synapsis is observed in heterozygous deletion, you will find the presence of deletion loop in the normal chromosome. You will find, I'll repeat it, you will find the deletion loop in the normal chromosome of the pair. This is the example of heterozygous deletion. So what happens in homozygous deletion? Now, here is again a line diagram showing homologous chromosome. You can see all the genes are matching. And then what happens? You find over here that homozygous deletion has taken place. In homozygous deletion, both chromosomes with same deletion. So what is lost? Which segment is lost? In both the chromosome of the homologous chromosome, you will find the segment which has the gene 6 and 7 is lost. So what is your observation, students? You can observe that both the chromosomes are of the same size. The genes are matching, centromere is matching. So in synapsis, you won't find the presence of any deletion loop. So students, when you study interstitial deletion, at the time of synapsis, if heterozygous deletion has taken place, then in that case, you will observe the presence of deletion loop at the time of synapsis. I hope you have understood. With the line diagram, I could make it clear how does deletion, terminal deletion takes place, and how does the interstitial deletion takes place. I also explained to you synapsis in synapsis, homozygous and heterozygous deletion. Synapsis in homozygous and heterozygous deletion. Now, these are the examples of deletion. I told you that the deletion, when it takes place, it depends on what is the size of the segment which is deleted. If it is a small segment which has been deleted, then in that case, it carries few genes. So in that case, the organism, it can survive, though it shows some symptoms because of the loss of the gene, but still the organism survives. Whereas if the deletion is a huge deletion, where the segment is very large, then in that case, it carries large number of genes and it may be lethal for the organisms. In human beings, deletion has been observed, and there are many symptoms which are seen in human, which is manifested as syndromes. There are some examples which I'd like to take in my next lecture, and today I will just mention them. So the examples of deletion is Wolff-Hirschhorn syndrome, Jacobson syndrome, Cri-du-Chat syndrome. Now, Wolff-Hirschhorn syndrome, it shows partial deletion of the short arm of chromosome number 4. So, students, you have done the karyotype, you have done the karyotype of normal male and normal female, and you have seen that there are 23 pairs of chromosome. So, we are talking about chromosome number 4. So if there is a partial deletion in short arm of chromosome number 4, it causes the syndrome Wolff-Hirschhorn syndrome. There's the second example. The second example is deletion of terminal part of the long arm of chromosome number 11. And this causes the syndrome which is called as the Jacobson syndrome in human being. And then, of course, the third type, which is the cat cry syndrome, or in French we call it Cri-du-Chat syndrome. It is the partial deletion of short arm of chromosome number 5. So students, I will be back with this lecture where I will be taking the various examples of deletion. I hope you have followed this lecture. Thank you. Thank you, and meet you for the next lecture.